Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, TuneIn Radio, Goodpods. Wherever you listen to podcasts, you can find Let's Talk Micro. As for our social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email, which is letstalkmicro@outlook.com. So please go ahead and subscribe to the podcast, download episodes, leave a review, any feedback if the app allows you to do so, and definitely any feedback, any suggestions, you can submit those via social media or via email. As always, thank you for your support. And if you haven't listened to the previous episode, please go ahead and do so. Great episode with Dr. Ryan Relich and Dr. Kenneth Gavina from the Indiana University School of Medicine. And they came to the podcast to talk about an article regarding Francisella and how they had a blood culture and they had an organism that was growing. And then they suspected Francisella, and then they sent it to their reference lab, which is their health department lab. And then it ultimately ended up being a subspecies of Francisella. So in the episode, they talk about biochemicals, morphology. They talked about being prepared, which is very important, and most of these organisms, that are potential bioterror agents, we thankfully, right, we might never see in our careers, and that's a good thing. But sometimes, we're so busy with our cultures and medical lab scientists, we have to get through a huge volume of cultures sometimes, or most times it seems like it. And then, sometimes, we get an organism and then we have trouble identifying it. We consult with someone, then maybe we consult with a supervisor, manager. And then next thing you know, we have 4 or 5 people that get involved. And they are potentially exposed to one of these organisms. So being prepared, keeping that knowledge and making sure we review, having some protocols for potential bioterror agents, have our policies in place. It's very important. So overall, it was a great episode, very informative. So please check it out if you haven't done so already. So in today's episode, we are going to talk about viruses. And, it was a great episode. I had 2 guests, and one of them was Dr. Ryan Relich from the Indiana University School of Medicine. And the other guest was Dr. Benjamin Pinsky from Stanford University School of Medicine, and he's also the editor-in-chief of the Journal of Clinical Virology from the American Society for Microbiology. So both of them, they're very knowledgeable. They are experts in the field of virology. So they came to the podcast to talk about emerging viruses and viruses of high consequence. And it was a great episode. So it's going to be 2 parts because, there's so much great information that I split it in 2. So part 1 is this episode, the one that is airing right now, and then part 2 will air next week. And then in part 1, Dr. Pinsky and Dr. Relich, they talk about viruses, going over the basics, DNA viruses, RNA viruses. They talk about the mode of entry. They talked about diseases. And then on the second part, they talk about, diagnosing them, laboratory testing, and the challenges. So overall, it was a great episode. And Dr. Pinsky, he talks about the Clinical Virology Symposium, which is actually happening from September 9 through the 12th of this year in West Palm Beach, Florida. So unfortunately, by the time this airs, it's too close to the symposium. But still, I am posting the link on the show notes, and go ahead and check it out. I mean, if you register remotely, you're able to access the content later for an amount of time. So, he said that, they have a good lineup. Him and Dr. Relich, they're doing a virology Jeopardy. So it seems that, it's going to be great information. I'm actually going to be going to it. I will be attending this year. And I'll be, posting about it, and I'll do an episode. Just like I did after Microbe 2023. And I'll be talking about the talks that I like, things that I saw that I enjoyed. So I'll be doing an episode about that. So stay tuned. But my apologies, when part 2 comes out, it'll be after the symposium. So you'll hear Dr. Pinsky talking about it. So I apologize for that. but I am including the link, so definitely go ahead and check it out. And if you are able to register remotely, go ahead and do so. There is some great content. So let's go ahead and listen to part 1 of this great episode about viruses with Dr. Relich and Dr. Pinsky. So on today's episode, we're going to be talking about viruses. It's something as medical laboratory scientists, we do testing. Maybe when we're going through the program, we don't go into them as much detail as we do bacteria, but definitely, very relevant topic. So with me today, I have two great guests that are going to be talking about emerging viruses and outbreaks of high consequence viruses. So I have Dr. Ryan Relich and Dr. Benjamin Pinsky. Doctors, welcome to Let's Talk Micro.
Thank you.
Yeah, thanks for having us.
My pleasure. So I think it'll be good. Let's just go ahead and start with a quick introduction to the audience. If you can tell them a little bit about yourselves and like what do you do and anything else that you want to add.
Yeah, absolutely. My name is Ryan Relich. I am infatuated with microbiology and have been since I was 10 years old. So getting to be a medical microbiologist and getting paid for it is something that, is great. So I guess you could say I don't really work a day in my life. But to that end, I am a medical microbiologist. I'm the medical director of the Division of Clinical Microbiology for Indiana University Health, based in Indianapolis, Indiana. I am medical director of a couple other laboratories here as well, director of a fellowship program for PhD folks interested in medical and public health laboratory microbiology. I'm an associate professor of clinical pathology and lab medicine at the School of Medicine, the Indiana University School of Medicine. And in addition to all of that, I also have an academic research laboratory in which I study emerging viruses. So I focus on risk group 2 and risk group 3 viruses that require BSL-2 and BSL-3 containment respectively. And yeah, that's pretty much me in a nutshell.
Thanks, Ryan. That was a great introduction. I did not come into microbiology or virology so early as you did. I thought I was very interested in laboratory medicine, but I really got obsessed with viruses during my clinical microbiology rotation and subsequently during the 2009 H1N1 outbreak, in which I brought in new testing and really got hooked on virology and the importance of viral diagnostics. I'm currently the director of the clinical virology laboratory for Stanford Healthcare and Stanford Children's Health. And my academic position is in the departments of pathology and medicine, as well as a courtesy appointment in pediatrics. Both medicine and pediatrics are in the divisions of infectious diseases, respectively, for the adult and children's groups. And all of that is in the Stanford School of Medicine, and I'm a professor in those positions. I do have a research lab which is primarily involved in the development and evaluation of novel diagnostics for infectious diseases, predominantly viruses, but also some other hard to culture organisms. So it's really a pleasure to be on here talking about emerging infectious diseases with you and with Ryan, and I think this is going to be a fun discussion.
Definitely a pleasure having you both. And like I was telling, Dr. Relich, I kind of just years ago, I saw him speak on something, and then I saw both of you doing the podcast at the ASM. So definitely, one of the for lack of a better word, the cool things of this podcast is, getting to connect and meet so people doing, great work out there in the field, and has like a little bit of a Hollywood feel for me, like meeting all these people that before maybe you saw their names on a publication or never actually spoke to them. So it's a great feeling. And I definitely can identify with what Dr. Roach said about the never working a day. It's just, I love what I do in this new journey. And, as a medical lab scientist, you can work in many areas, and I have tried to be objective and I have worked in them. I've done hematology, blood bank, but it's just, micro has that pool that it's just, and definitely about 2 years ago, I said, this is it. I'm throwing myself in completely. And while those areas are great, for me, it's micro. So I'm definitely all in.
So I should also mention that Ryan and I kind of got thrown together to do clinical virology Jeopardy, which is a staple of the clinical virology symposium. We took over from Alex Valsimakis, who had done it for many years and did an amazing job. And so we've been doing it for the last several years, of course, with a COVID-2 hiatus. But we are reprising that later this year in September at the Clinical Virology Symposium. So we enjoyed chatting with each other, and hopefully this won't get too out of control.
Yeah, the banter sometimes gets a little bit carried away. So whenever we derail, just re-rail us and,
Yeah, exactly. Keep us on.
Push us down the tracks.
I mean, we love virology. So, hopefully this is as entertaining for the listeners as it is for us.
Yeah.
Yeah, right. So definitely, looking forward to this. So let's go ahead and just talk about some emerging viruses. And I think it's always good for the audience, like I mentioned at the intro. Sometimes when we're studying or going through the program and things like that, maybe we don't touch on them as much. Maybe we stick to some classics, some very short things. It's a time thing, and you don't have time. You're preparing for your boards, so definitely here we can go a little bit more detail and hit some points that are very important. So let's go ahead and talk about some. We hear about DNA viruses versus RNA. How do they? What's the mode of entry? Reservoirs. How do we treat them? So all those main points. So let's go ahead and start.
Sure. Well, who would you like to talk about this stuff? Ben and I are equally passionate. Maybe we could just start talking at the same time. And
No, yeah. Why don't you go first, Ryan, and then I'll jump in with my favorite viruses, and I'm sure we'll have a lot to talk about.
Yeah, absolutely. So, all right. Well, let's turn the clock back to Basic Virology 101. So there are 7 different types of genomes among viruses out there, comprising either DNA or RNA. And the format, if you will, of those genomes differs from virus group to virus group. So, for example, if we start with DNA viruses, you have single-stranded DNA genomes, you have double-stranded DNA genomes, and there are others that are called gapped double-stranded DNA. A good example of that is the hepatinovirus or the hepatitis B virus. And so, in addition to that, there's this whole cadre of RNA viruses that comprise the other 7 genome types. There's the double-stranded RNA viruses, things like reoviruses and rotavirus. There are what are referred to as the minus sense single-stranded RNA and the plus sense single-stranded RNA viruses. And there are a number of emerging viruses, if you will, that are essentially members of those 2 broad categories of viruses. And the last genome type is the RNA that employs reverse transcription in order to make a DNA intermediate that is then used for other things. And good examples of those would be lentiviruses like HIV. And so many of the emerging viruses, if you will, that we've seen over the past several years that have emerged, the majority of them are RNA viruses. And so folks then ask themselves, well, why RNA viruses? Why not DNA viruses? Well, that answer has a couple of different parts. Number one, DNA viruses do emerge and they do cause significant problems. Case in point, most recently, monkeypox virus emerged, and it spread throughout immunologically naive populations throughout the world by close contact with infected humans. And that chain of human-to-human transmission started, much like all other viral infections that we know of these days, it started with some sort of animal host. whether it be a reservoir or some intermediate. And then the virus, what's referred to as spilled over. So there was a spillover event that led to the virus being, um, introduced into the human population. And so RNA viruses, though, account for most of the ones that make the headlines these days. And there's a couple of different reasons for that. The chief among which, I think, and I could be wrong about this, maybe there's other learned people that know much more than me. But one of the things that RNA viruses, a trick that they have up their microscopic sleeves, is sort of their breadth of genetic diversity. And so with, for example, SARS-CoV-2, the virus that's associated with COVID-19, it and other RNA viruses exist within cells during the manufacturing process, or what's called the infectious cycle, and what's called a quasi-species. And a quasi-species, it's all SARS-CoV-2, but there are a number of genetic variants that are present within the population of viruses that's produced within a host cell. And so many of those viruses can have mutations that may not affect their infectivity or replication. Others can have deleterious mutations that essentially, shoot them dead in the water. And others may have adaptations that lead to something that benefits them. And so perhaps in a very large pool of viral progeny, there's only maybe 100 or so that are capable of going on to infect cells, and perhaps some of those are particularly well adapted at infecting human cells. So, that genetic diversity that is a consequence of their just normal being is really lends itself quite well to something that would eventually then make it much more fit for emergence in the human population. And so with regard to mode of entry, there's a couple of different ways that these viruses can get into people.
Ryan, before you go on to mode of entry, can I just jump in and say the diversity, I think, is a really important part of this, and at least some of the mechanistic reasons for this is that the RNA-dependent RNA polymerases that these viruses have make a lot of errors. So they are not high-fidelity enzymes. Many do not have any sort of proofreading activity or have minimal. I think I'm correct on that, Ryan. You can correct me.
Yeah, you are correct.
And so the virus is replicating and has and makes these mistakes. And as Ryan said, many some don't do anything, some are deleterious, but others give them some sort of selective advantage and may make them more pathogenic, more easily transmissible, and so forth. So they have this error-prone activity, as well as if viruses replicate to high levels, there's the opportunity to make errors. And so these two things combined then have there's these possibility that you can create viruses in a reservoir species that can then, as Ryan said, spill over into human populations if there's the right interactions. And so that's where we get concerned about these viruses, and also why this sort of genomic monitoring that has become so popular with SARS-CoV-2, but why that's so important for a lot of these emerging viruses, so we can see, what's going on with these viruses. Are they generating, are the mutations that are being generated, do they have the potential to cause problems? So sorry I interrupted. I just want to jump in there and
no, that's very important. So,
and you should go on, because then the next step is what you were talking about, like how these transmit, how do they get into humans? What are those risk factors that happen?
Yep. Yeah, absolutely. So the mode of entry into human hosts can be one of many possible ways. For example, spillover events that occur, consequent to bushmeat gathering or hunting, is typically exposure to blood and consumption of meat that may contain viruses. Vector species such as ticks, mosquitoes, biting flies, and other things are really good ways by which viruses can travel from one animal to another. And there's, a variety of ways in between that the viruses can also get in. Direct inhalation of infectious particles. A good example of a virus that does pretty good at getting into human hosts through breathing them in terms of emerging viruses, are hantaviruses, orthohantaviruses. So they're shed in the excrement of rodents. And even though that excrement can dry on a surface and be dried for a while, and in the act of disturbing it and sweeping it up, you create basically a microparticulate aerosol that you can breathe in, and it'll land on your mucous membranes, and some of it will get swallowed, others will get taken down into the small air spaces within your lungs, giving the virus then an opportunity to bind to susceptible and permissive host cells. And so really acquisition and entry are tied together that way. There are, in some instances, multiple potential ways that a virus can spill over into the human population, whether it's directly through the reservoir exposure to a reservoir species or an intermediate host. But regardless of what happens, if the virus has the ability to form a stable interaction with the new host, then you can get these chains of transmission from person to person. And along the way, the viruses can adapt. So SARS-CoV-2 is an example of a virus that we used to call a zoonotic virus, but now, after 3-ish years or more of sustained human-to-human transmission, that virus has done a pretty good job at adapting to the spread throughout humans, and so we call that a human virus now. But there's many other examples of zoonotic viruses that occur in the human population consequent to a spillover. Ebola viruses are an example, Marburg virus, Nipah virus, and a handful of others, just, sort of at the top of my head.
Yeah, and certainly you mentioned earlier monkeypox virus causing mpox disease also was thought to be a zoonotic due to zoonotic exposures, and now clearly has occurred through person-to-person close contact in certain populations.
Yeah, absolutely. And there's a lot of animals that are implicated in nature to human, if you will, transmission of these viruses. And rodents and bats get a lot of press with regard to that because rodents account for about 40% of the mammalian species diversity, and bats account for another 20% of the mammalian species diversity. Of the 1,200 or so species of bats, each of those bats may harbor viruses that are only found in those bats, and sometimes they spill over into the human population. And most of the time, it appears that those viruses probably do nothing. There may be a brief infection that gets burned out very quickly. Perhaps the host, the human cells are not susceptible, they're not permissive to infection. And so the virus doesn't get an opportunity to replicate and then go from person to person. Every once in a while, though, a bad actor will emerge, and viruses like rabies virus and Nipah, Hendra, Ebola viruses, Marburg viruses, various beta coronaviruses. Those viruses then have, unfortunately, the ability to lead to long chains of human-to-human transmission, as long as the circumstances that dictate that human-to-human spread are maintained. But you need to interrupt that chain of transmission then to put an end to those generations of viral spread. Does that answer your question, or is that effectively tap dance around it, Luis?
No, yeah, it does, yeah. Thank you. Definitely a lot of good information.
And I don't mean to villainize these animals, because they don't care about us per se. They're not deliberately giving us viruses. A lot of these exposures are a consequence of our own actions. People are really good at messing up ecosystems and displacing native species, and effectively by all of that, interrupting these enzootic cycles of viruses in nature. And then, one thing that's really dramatic is the human population growth over the past, 500-600 years. The human population was pretty small, I mean, compared to now. And in the span of 500 or 600 years, just billions of people grew up. Our reproductive potential went through the roof, and now we're, in every corner of the earth, exploiting resources, displacing natural populations of animals, leading to increases in the number of incidents of exposure to these, wild animals and their wild viruses.
And the other thing to point out there is that in many of these reservoirs, the viral infections are asymptomatic. Like the viruses and the host have come to an equilibrium such that the viruses are just very effective at maintaining their genomic material, and they don't really do much to the host. And then we step in there and our immune systems and the viruses themselves cause a lot of problems.
Okay, so as you have mentioned, we have gone over viruses, DNA, RNA, how do we acquire them, the reservoir. Maybe can you touch a little bit on some, definitely a lot of information, but maybe touch on some diseases and treatment.
Sure. Ben, do you want to talk about diseases and treatment?
I mean, it's a broad category. You want to go for it? I can jump in.
Yeah, I'll do the hemorrhagic fevers, and you can do the non-hemorrhagic fevers. How about that? Yeah, so there's a whole array of viruses out there that are associated with this syndrome referred to as hemorrhagic fever. And so not every person that contracts these hemorrhagic fever viruses, though, develops hemorrhagic fever. It's really the sort of the exceptions to those that get the most media attention, especially considering they're high consequence pathogens. But chief among those that cause these severe acute infections that can have high case fatality rates are viruses like the filoviruses, Marburg and Ebola viruses. And real quick, a word about nomenclature here. So whenever we use the word Ebola virus as two separate words, that is specific to what's called Zaire Ebola virus, which is a species of the genus Ebola virus. There are three other Ebola viruses, all one word. Three other members of the genus that include Sudan virus, Thai forest virus, and Bundibugyo virus that also cause a disease, Ebola hemorrhagic fever. And so there's also Marburg virus, which is associated with hemorrhagic fever. And there are a number of arenaviruses, like Lassa virus, Lujo virus, the South American hemorrhagic fever viruses like Guanarito, Junin virus, Machupo, Chapare virus. Here in the United States, there's even probably more than one, but at least one that I know of that I have in the laboratory. It's called whitewater arroyo virus. That's a mammarina virus that appears to be reservoired by cotton rats, which are found throughout the desert Southwest, so New Mexico, Arizona, California. And there have been cases of human disease linked to exposure to those animals and subsequent infection with whitewater arroyo virus. There are the hantaviruses, which are associated with hemorrhagic fever and renal syndrome. Typically, what are called the old world hantaviruses, like Hantan virus and Sol virus and Pumala virus, and various flaviviruses like yellow fever virus that is associated with hemorrhagic fever. And there are a bunch of others, non-hemorrhagic manifestations, including respiratory disease and the central nervous system disease. And Ben, if you want to talk about those for a minute, go for it.
Well, it's just you mentioned the hantaviruses, and we were just talking about with my residents today, Sin Nombre virus, of which there was about, I don't know, 7-8 years ago, maybe a little longer, quite a large outbreak in Yosemite National Park, not too far from us here. But certainly all of those viruses you mentioned are of concern and can cause severe illness if individuals are exposed. One thing that you didn't talk about was, the therapeutics for these viruses, of which the options are extraordinarily limited. It's primarily supportive care for folks that are infected and have severe clinical illness. I'm not aware that there are specific antivirals for any of those. Folks try a variety of things that may impact viral replication, but typically this is all supportive care. So viral therapeutics are something where a lot of work needs to be done to identify therapies that are effective at preventing severe clinical manifestations. The main ones, as you know, are there have been several developed for SARS-CoV-2 that have modest effects and are, modest effects at preventing infection, but can reduce the severity of illness. And those include protease inhibitors, as nirmatrelvir, also known as Paxlovid, and remdesivir, which is an RNA-dependent RNA polymerase inhibitor. And for a lot of these emerging viruses, though, we don't have drugs. Flu, we have neuraminidase inhibitors like Tamiflu or oseltamivir, and some newer RNA-dependent RNA polymerase inhibitors. But for a lot of things, really, the development for antiviral drugs has been primarily focused on chronic infections. Of course, first HIV, which has been a huge success, I would argue, and more recently hepatitis C, which can now be cured with antiviral medications. But a lot of these emerging viruses, as I said, are really, we're limited in our arsenal to treat patients that have become infected. And really the approach is, and Ryan can talk more about this, is to develop vaccines. So should there be a spillover, we can limit its spread. And so that's been the case for filoviruses, for Ebola and Marburg, certainly. And I think that's the approach that's being taken for many of these infections, that if there is spillover, we can really limit its extent by vaccination.
Yeah, absolutely. And there's a specific approach that's taken, referred to as ring vaccination. For Ebola virus, the Zaire Ebola virus, there's a couple different vaccine platforms that exist. The one that is FDA approved in the United States, which uses a recombinant vesicular stomatitis virus that expresses the Zaire Ebola virus glycoprotein. That is what's available, to many places in Africa whenever they need it. So that vaccine gets introduced, and they try to identify people that were contacts of specific cases. Then they vaccinate, the contacts, they vaccinate the contacts of the contacts, essentially creating this concentric ring of vaccinated individuals around each case, in an attempt to sort of stamp out the transmission of that virus. And that type of approach, for years has been what folks, biopreparedness folks, have thought would be good to handle smallpox in the event that smallpox was deliberately introduced as a biological weapon into the human population. They would find individuals and they would ring vaccinate around those folks. And so that type of thing is useful, and for many reasons, for curbing the transmission of these things, these viruses. And we haven't even talked yet about respiratory viruses and the neurotropic viruses. I mean, there's a whole laundry list of viruses. I only went with hemorrhagic fever because that's the one that everybody knows about, right? But influenza viruses, like avian influenza viruses, those are emerging viruses. Even vaccine-derived poliovirus type 2 is emerging, in the sense that, in the United States, in New York, for example, there's probably thousands of these cryptic cases of vaccine-derived poliovirus type 2 infections, but it's only one in a few hundred or so people that develop signs and symptoms of disease. Meanwhile, the rest of the population who's infected with those viruses, perhaps with either mild or no symptoms at all could be shedding the virus into their feces. And then, we can detect it through something that's been employed now quite a bit for SARS-CoV-2 surveillance, which is wastewater surveillance.
Yeah, absolutely. That is a new weapon in the arsenal of public health to identify potential emerging pathogens by what's in the wastewater. Of course, that depends on the pathogen being shed at high enough levels in waste. um, but certainly that's been used globally for polio surveillance, in particular vaccine-derived polioviruses. The polio vaccination, because this is oral polio vaccine, which is live attenuated and can undergo phenotypic reversion. So once this once the vaccine strain, if that is able to persist in a population, it picks up new mutations that then allows it to again cause polio, which is really disconcerting. There's new vaccines now where very clever folks have basically used molecular biology techniques to make it such that it's more difficult for the virus based on the sequence to mutate known residues that result in phenotypic reversion. So these are these novel or new OPV2 vaccines, and they're working on it for the other serotypes, even those are not currently circulating. But that's a really interesting approach, although it'll probably take a few years before these vaccine-derived polioviruses are eliminated. And certainly there's still a handful of countries where wild polio is still circulating. Afghanistan and Pakistan, I believe, are the two remaining countries that still have not eradicated polio. I think Nigeria is considered eradicated at the moment. Yeah, so that was a sidetrack to poliovirus, which is just totally fascinating and something we really don't think about very much in the United States because it's been eliminated here for decades, and everyone is vaccinated with inactivated polio. So the live attenuated polio is really imported, and then with waning immunity, people get infected and it continues to transmit.
And that, my dear audience, is the end of part 1 of the Emerging Viruses episode with Dr. Benjamin Pinsky and Dr. Ryan Relich. I hope you enjoyed it. As always, I enjoy sharing this information with you. So please stay tuned. The second part will be airing next week. And if you're stopping by the Virology Symposium, and if you're attending this year, please go ahead and stop by if you see me. I'm always looking forward to meeting people, connecting, and learning. As always, continue bringing that passion to what you do. It's so important. We do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.